Hello buddies, welcome back to the channel BioPulse, and this is Chandrika. Today's our journey of day 34 of the 40 days journey, and we are discussing the chapter Biotechnology Principles and Processes. Biotechnology actually is a very fascinating field that combines biology and technology. That's what the name biotechnology means. To develop what? To develop useful products and solve real life problems. From producing life saving medicines like insulin, that is creating genetically improved crops, and biotechnology plays a very major role in modern science and then healthcare. In this chapter we dive into the core principle of the genetic engineering, where DNA is manipulated to achieve desired traits. Okay, we will understand how scientists use tools like restriction enzymes, vectors, and host cells to transfer genes from one organism to the another one. And this chapter also explains important techniques like recombinant DNA technology, PCR, that is polymerase chain reaction, and gel electrophoresis. And you also will explore the step-by-step process of the gene cloning in this chapter. Okay, and for the if I talk about the NEET, for the sake of the NEET, you may expect 1 to 2 questions from this chapter, and this is very foremost chapter, I can say, which will be very must interesting, which is best interesting chapter, I can say. So let's dive into the chapter, that is biotechnology principles and processes. Here is the first question. EFB stands for option A, English Federation of Biotechnology, option B, European Federation of Biotechnology, C, English Federation of Biotechnology, D, European Federation of Biotechnology. So here the right option is option D, European Federation of Biotechnology. So the next question is, two core techniques that enable birth of modern biotechnology are A, physical and biological engineering, B, bioprocess and genetic engineering, C, molecular and cellular genetics, and then D, none of this. So here the right option is the B, bioprocess and genetic engineering. What actually the genetic engineering? This involves technique to alter the chemistry of the genetic material, that is the DNA or else the RNA, okay, to introduce them into host organism that can be changed their phenotype. Okay, that means the external characteristic. What about the bioprocess means? What actually this one? This involves the maintaining a sterile environment in the chemical engineering process to allow a large scale growth of the only desirable microbes or a eukaryotic cell of that, I can say. So this is about these two engineering process. Next, third one, question number third, biotechnology use techniques to alter chemistry of protein and lipid, protein and RNA, lipid and DNA, RNA and then DNA. So here the right option is option D, RNA and DNA. Next, fourth question. In chemical engineering process, it is important to maintain A, maintain micro-free environment, B, micro-full environment, C, sterile environment, and then D, more than one option is right. So here the right option is D, because more than one option is right. Which one are right? Maintain micro-free environment and sterile environment. Next, fifth one. Unique condition of genetic setup is naturally provided by unique combination, sorry, not condition, combination. Unique combination of genetic setup is naturally provided by sexual reproduction, asexual reproduction. Biotechnology more than one option. So here the right option is sexual reproduction. Naturally creates a unique combination of genetic setup through the process like crossing over during the meiosis, okay na, or else the random fusion of the gametes, we can say. This varies in the basis of evolution and then adaptation. That's what this combination is unique. Next, sixth question. All the genetic changes occurring naturally are harmful to organism and its population, beneficial for organisms and its population, not harmful for the organism and its population. D, both A and C. So here the right option is option A. All genetic changes occurring naturally are harmful to organism and its population. Like what? Naturally genetic changes means mutation, I can say, isn't it? Most random changes to the it will leads to what, like finely tuned genetic system are disruptive, isn't it? Likely to be determined to the individual or else the population over fitness. That's what this option is proper one. Next, genetic information is preserved by genetic information. Seventh question it is. Genetic information is preserved by A, sexual reproduction, B, asexual reproduction, C, both of these, and D, none of this. So here the right option is option B, asexual reproduction, because this process involves a single parent and doesn't involve the fusion of the gamete. As a result, what happens? The offsprings are genetically identical to the parents, isn't it? We can call it as a clones, isn't it? Effectively preserving the genetic information without any kind of the changes. But when we talk about the sexual reproduction, what happens? Here fusion of the male and female gametes is going to take place, which leads to gene recombination and segregation also. So this process creates variation rather than the preservation. So here the next question is question number 8. When a piece of DNA is transferred to an alien organism as it is, A, it will multiply itself, it will not be able to multiply itself, and then C, it will be present in progeny cells of the organisms, and then D, both A and C. So here the right option is B, because it will not able to multiply itself. That's what the piece of the DNA is transferred to an alien organism because of the lack of the oversight. Okay. Next, ninth question. Chromosome replication is initiated at a gateway of replication, a specific RNA sequence, B, origin of replication, a specific RNA sequence, C, path of replication, a specific RNA sequence, none of this. Here the right option is option B. Next, 10th question. For alien DNA to replicate, it needs to be a part of A, chromosome without origin of replication, no. Mitochondrial DNA with origin of replication site, no. Chromosome with origin of replication site, yes, this one is correct. So the right option is C. Next, 11th one. Plasmid is A, autonomously replicating extra chromosomal, yes, this one is right. Non-autonomously, no. Autonomously replicating chromosomal. It's not a chromosomal, it's a extra chromosomal. It's autonomously replicating extra chromosomal because it is a very small, typically circular double-stranded DNA, I can say, which is exists separately from the main bacterial chromosome. If I say this is the bacterial body, and if it is having its main genetic DNA is here, it has its one more extra chromosomal DNA which is almost circular, that is autonomous, and autonomously replicating means it can replicate itself. So this one is said to be what? Plasmid. So the right option is A. 12th one. Plasmid is linear RNA, B, circular RNA, C. Linear DNA. D. Circular DNA. So here the right option is it's a circular DNA. It's actually extra chromosomal circular DNA molecule that is physically separated from the chromosomal DNA and replicate independently. They are the most commonly found double-stranded circular DNA in the bacteria. Next, 13th one. First recombinant DNA involved native plasmid of A. Escherichia coli, B. Salmonella typhimurine, C. Streptococcus pneumoniae, D. Clostridium botulinum. So here the right option is option B, Salmonella typhimurium. So the first recombinant DNA was constructed in the year 1972. In the year what? 1972 by Stanley Cohen and Herbert Boyer. Okay, they achieved this by linking a gene encoding antibiotic resistant with the native plasmid what? Salmonella typhimurine. Next, 14th one. First recombinant DNA was made by, just now I said, isn't it? Cohen and Boyer in the year 1972. Herbert Cohen, no, not the Herbert, it's a Stanley Cohen, Herbert Boyer, but not the 1992, it's a 1972. Stanley Cohen and Herbert Boyer in 1972. Yes, this one is the right one. Next, 15th question. The recombinant DNA was made A. Before discovery of DNA cutting restriction enzyme. No. After discovery of DNA cutting restriction enzyme. Yes, that is recombination DNA, that is rDNA, was made fundamentally on the ability to cut the DNA at a specific sequence to insert new genetic material in that. Okay. And restriction enzyme is also called as molecular seizure, where the discovery that made this possible, that is cutting of the DNA. The first recombinant DNA molecule was created in the year 1972. Just now I said about that is by Cohen and then Boyer, isn't it? Next, 16th one. The plasmid DNA linked with cut piece of DNA acts as A. host, B. as a vector, C. medium to transfer the DNA piece, and then D. more than one option is right. So here the more than one option is right because it acts like a vector and also medium to transfer the DNA piece also. So both are right about the plasmid. So more than one option is right. 17th question. Linking of antibiotic resistance gene with the plasmid is done using the enzyme. Linking means what? Like glue. Antibiotic resistant gene with the plasmid is linked by the help of what? Ligase enzyme. It is called as molecular glue. This enzyme is used to join the DNA fragments together by forming phosphodiester bonds. Next, 18th one. The plasmid joined with the required DNA of interest is transferred into dash by Boyer. A. Escherichia coli, Salmonella typhimurine, Streptococcus pneumoniae, Clostridium botulinum. So here the right option is option A. Question number 19. The key tools for recombinant DNA technology are A. restriction enzyme polymerase, hydrolase, and then vector, and then B. recognition enzyme polymerase, ligase, and vector, and C. restriction endonuclease, polymerase, ligase, and vector. D. restriction enzyme polymerase and dehydrogenase vector. So here the right option is option C. Restriction endonuclease is for molecular seizure. These enzymes cut the DNA at a specific recognition site, whereas polymerase, it is used to synthesize new strand, and ligase is for a glue, like molecular glue. It acts like a molecular glue. And vector, these are for the DNA molecules used as a vehicle to carry a foreign DNA into the host cell. So these all are the tools we needed. Next, 20th question. In 1963, two restriction endonucleases were isolated in E. coli that restricted growth of the bacteriophage by A. cutting DNA, B. adding methyl group at DNA, C. removing methyl group to DNA. More than one option is right. So here the right option is option D. More than one option is right. That is because adding methyl group to DNA and cutting DNA, these two. 21. The first restriction endonuclease was HIND3, HIND2, HIND1, HIND4. So here the first restriction endonuclease was HIND2. HIND2 was first restriction endonuclease to be isolated and characterized. It was discovered in the year 1970. In the year 1970 by Smith and his colleagues. Next, 22 question. E. coli comes from genus Iconia, species coli, genus Echinus, species Crispus. So here the right option is option B, species coli. The name of the restriction enzyme E. coli is derived from the scientific name of the bacterium, that is Escherichia coli RY13. So here the right option is option B. Next question, question number 23. Recognition sequence is A. specific sugar sequence in the DNA which is recognized by the restriction endonuclease, and then B. specific protein sequence which is recognized by the restriction endonuclease. C. specific lipase sequence which is recognized by the restriction endonuclease. Specific base sequence in the DNA which is recognized by the restriction endonuclease. This one is the right. Actually, recognition sequence is the is a specific base sequence, I can say, in the DNA that is recognized and bound by a restriction endonuclease. Okay, that's what it is. Recognition sequence it is said to be. It is a specific base sequence in the DNA. Next, 24th one. The convention for naming restriction endonuclease is the convention for the naming restriction endonuclease is first two letters from the genus and third from species of prokaryotic cell. No, first two letters from the species and third from the genus of. No, first letter, yes. First letter comes from genus and second two from species of the prokaryotic cell from which they were isolated. Yes, this one is right. From first letter comes from species. No, it must be from the genus. So here the right option is option C. See here what happens. First letter will be from the genus and next two letters derive from the species. If I go with the example, last we had this question. No, this one, E. coli. Let me explain that one. E, in this E. coli, E. core, this one. Here E. core 1. Here E is from Escherichia, that is a genus. Okay, and co, 2 letters from species, that is coli, and ry13, it's a strain, and then this one is first enzyme isolated from this strain. Okay, so here first letter comes from the genus, second 2 letters comes from the species of the prokaryotic cell from which they were isolated. So here it is isolated from the Escherichia coli. Next, 25th question. Roman number indicate. What the Roman number indicate? Order in which enzyme were isolated. Yes, of course, this one itself is the right. Next, 26. Restriction enzyme belongs to exonuclease, endonuclease, both, and then none. So here it is belongs to endonuclease, that is B option is right. What actually endonuclease, exonuclease? Endonuclease means these cut the DNA at the specific points within the DNA molecule, and whereas exo, these remove nucleotide one at the time from the ends of the DNA strands. Okay, they either 5' end or else 3' end. So endonuclease means they cut the DNA at the specific points within the DNA. They will be within, that's what the word endo is given. Exo means at the ends, either 5' or else 3'. Next question, that is question number 27. Endonuclease cuts DNA from specific position within the DNA. No. Ends of the DNA. Yes, this one itself is right. Either 5' or 3' ends. This one, this exo means what? Outside. Endo means inside. Next question, question number 28. Restriction enzyme recognize A. Palindromic sequence of the nucleotide in the DNA, palindromic sequence of nucleoside in the DNA. He said nucleoside. This one is also nucleoside. That is not nucleoside, it's nucleotide. Palindromic sequence of nucleotide in DNA, palindromic sequence of nucleotide in DNA. So here the right option is D, because in this is, sorry I spelled wrong, paleondromic is given, so this one is wrong here, and among these two nucleoside is wrong. So palindromic sequence of the nucleotide in DNA. What is palindromic sequence? Palindromic sequence is like a sequence of the base pair that reads the same in the 5' direction, 5' to the 3' direction also, and 3' to 5' direction also. That, for example, if I say ECOR recognize the palindromic sequence of GAATTC, like 5' from 5' GAATTC 3'. This is from 5' to 3'. If I'm talking about 3' to 5', C T T A G. This is 3' to 5'. So this is called what? Palindromic. Okay, this is palindromic sequence. ECOR recognizes this sequence to cut. Next, 29th one. Which of the following enzyme is used to cut DNA of at a specific sequence? DNA ligase? No, it is for joining. Restriction endonuclease is right. And then DNA polymerase, it is used to synthesize the new DNA strand by adding nucleotide. And reverse transcriptase, it is enzyme used to generate the complementary DNA, that is a cDNA from the RNA template. So this is also wrong. 30th one. Which of the following is palindrome? Which of the following is palindrome? GAATAC and GCTT. No, this one is wrong. GATATAC. No, this one is also wrong. GAATTC. CTTAAG. Yes, this one is right. So the right option is C. Next question, question number 31. Restriction enzyme cuts DNA between same two bases on opposite strands in the center of the DNA sequence. No, not in the center. Between same two bases on opposite strands a little away from the center of the DNA sequence recognition. This one is right. Next, 32. Same restriction enzyme produce A. Same kind sticky ends joined using endonuclease. No. Different kinds of sticky ends joined using ligase. No. Same kind of sticky ends joined using ligase. This one is right. See, here he is given endonuclease, so this is wrong. And different kind is wrong. Same kind of sticky ends joined using ligase. Next one. Identify correct labelings. In the given figure, identify the correct labelings. So here 1, 2 and then 3 is given. This is 1, this is 2 and then this is 3. This is recombinant DNA technology. This is the foreign DNA. This is foreign DNA. And this is the vector plasmid, the circular DNA used to carry the foreign DNA, isn't it? This is the source of the DNA containing the gene of interest, whatever the gene of interest we have, which is cut into the fragments. And then this DNA is carried by the vector, that is vector plasmid. And next it is going to be recombined, that is recombined. This is the recombinant DNA. So here 1 is foreign DNA, 2 is vector plasmid and then 3 is recombinant DNA. So the right option is option B. Next question, 34th one. The process of transformation is taking place when bacteria replicates and makes copies of rDNA with it. Next, bacteria picks up rDNA. Yes, foreign gene is added to cloning host prokaryotic cell. More than one option is right. Here the right option is option B. 35th question. Separation and isolation of the DNA fragments from the concept. 35th. The technique used for separation of the DNA fragments are gel electrophoresis. Yes, of course, this one is the right. Actually, this is a standard laboratory technique, I can say, specifically used to separate and isolate DNA fragments based on size and electrical charge, based on the size. And when we talk about the DNA fingerprinting, it is the process uses electrophoresis. It is a broader forensic method for the identification rather than just a technique for a general fragment separation. It won't separation. And when we talk about PCR, that is polymerase chain reaction, this technique is used to copy the DNA. And about DNA cloning, this is the process of making multiple identical copies. So here technique used for the separation of the DNA fragment is gel electrophoresis. Next, 36th one. DNA fragments are negatively charged, positively charged, neutral, none of this. DNA fragments are negatively charged because of the phosphate group, okay, which is present in their chemical backbone. Next, 37. In gel electrophoresis, DNA are forced to move towards A. Anode under magnetic field. B. Cathode under magnetic field. C. Anode under electric field. D. Cathode under electric field. So it moves move towards anode under the electric field. 38th one. Matrix used in electrophoresis is A. Ethidium bromide. B. Agarose gel. C. Neutral polymer extracted from the seaweeds, and then D. More than one option is right. So here more than one option is right. That is matrix used in the electrophoresis is agarose gel and then natural polymer extracted from the seaweeds, because this statement is defining about the agarose. Next, 39th one. Ethidium bromide is used to stain because DNA fragments are visible without staining. DNA fragments are not visible under staining. DNA fragments are not visible without staining. DNA fragments are visible under the staining. So here DNA fragments are not visible without staining. So you can use ethidium bromide to stain the DNA. 40th one. Stained DNA is exposed to visible light, UV light, IR light, radio wave. So it is exposed to UV light. Next, 41. Color of DNA visible under UV light after ethidium bromide stain is blue, black, orange, green. Here the right option is C, orange. 42. The extraction of separated bands of DNA from agarose gel are dilution, elution, dilution. So here the right option is option C, elution. That is a specific process where the DNA bands is cut out from the agarose gel and DNA is extracted from the gel pieces. So this is the gel electrophoresis structure. 43rd question. So what is identify labels correctly? 1, 2 and then 3 here. What is one? First one is well, and then second one is largest DNA strands, because they are towards the well and they will be of small. These are the small bands, isn't it? So here third is given small. So well, largest DNA bands and smallest DNA bands. The right option is B. Now questions from cloning vector. 44th question. Plasmids in bacterial cells replicate A. Depending on chromosomal DNA, independent of chromosomal DNA, depending on extranuclear DNA. More than one option is right. So here the right option is option B, independent of chromosomal DNA. Plasmid in a bacteria cell replicate, it is independent of chromosomal DNA. That is not at all depend on the chromosomal main DNA. 45th one. Bacteriophage. It replicate independent of other organisms. Replicate inside the bacterial cell controlled by the chromosomal DNA of the bacteria. Replicate inside the bacterial cell autonomously. More than one option is right. So here the right option is option C, replicate inside the bacterial cell autonomously. Bacteriophage and plasmids are used as a cloning vectors because they have the ability to replicate within the bacterial cells autonomously. This means they replicate independently. Next, 46th one. Bacteriophage serves as a dash in biotechnology. Just now I said this serves as a vector. Now this is the figure for the question 47 to 53. 47th question. Identify BAMH in the given plasmid figure. Where is BAMH? So identify BAMH. So here let me tell you what these all are things. 1 and 2, this one and then 2 are this is E core and this is hind 3, and here third one is BAMH 1. Okay, and fourth one is a SAL 1, and this one fifth one is PST 1, whereas sixth one is PVU 2, whereas A is ampicillin resistant gene, B is tetracycline resistant gene, whereas E is oricide, that is origin of replication, and then D is, that is ROPE, codes for the protein involved in the replication. So here he is asking about BAMH1, so we will go with the option C. Next, identify antibiotic resistant gene in the figure. Antibiotic resistant. Which one is antibiotic resistant? So here, identify antibiotic resistant gene in the figure. So here the right option is ampicillin resistant gene. Next one, identify E core 1 in the plasmid. I said about that. E core 1, where in which E core 1 is there? Second one, isn't it? Second one is E core 1. Where is 2? So the right option is D. Next, 50th question. A and B in figure are ampicillin and tetracycline. So the right option is option A itself. 51. ROPE, codes for I, and is shown in the figure by 2, isn't it? So here the right option will be protein involves in the replication. So this one itself is the right, D. Next, 52. Ori means dash and is shown in the figure by. Ori means its origin of replication, and in the figure it is shown by C, isn't it? Origin of replication and C. Let me see. Ori, yeah, it is origin of replication, it is shown in C. So we will go with the option D, origin of replication, and it is shown in the figure by C. 53. Identify PVU2 in the given figure of the plasmid. PVU is at let me take you there. So here PVU 1 here that is at the place of sixth one, isn't it? This is the PVU. So we will go with the option sixth. So option C is right here. Next question, 54th one. Which of the following is correct? Any piece of DNA linked to Ori gene will be replicated. Yes, this one is correct. Number of replication copies is under control of recognition site. No, it is the number of copies is controlled by Ori, isn't it? Number of replication copies in under control of recognition site. No, this is wrong because it is controlled by Ori. that is origin of replication. That's what it is incorrect. Vector should not be chosen based on the number of copies supported by it. No, vector should be chosen based on the number of copies it supports. Should not, he is given, so this one is wrong. So the right option is option A. 55. Transformants include cells which have picked vector with foreign DNA ligated to it. Yes. Cells which have picked up vector without foreign DNA ligated to it. This one also right. So both A and B. Next, 56th one. Recombinants are A. Cells which have picked vectors with foreign DNA ligated to it. Yes, this one itself is right. Next question, 57th one. Which is true about recombinant and transformant? All transformants are recombinants? No. All recombinants are the transformants? Yes. 58th one. Normal E. coli cell carries resistance against antibiotic ampicillin, tetracycline and kanamycin. Next, doesn't carry resistance against antibiotics, ampicillin, tetracycline and kanamycin. Carries resistance against ampicillin but not tetracycline and kanamycin. Carries resistance against tetracycline but not the ampicillin and kanamycin. Here the right option is option B. 59th one. In order to link alien DNA, vectors need to have a dash recognition site for commonly used restriction enzyme. So here very few, preferably single, many, more than one option is right. So here preferably single. Next, 60th one. Assertion and reason type of the question. Here assertion is given, vector should have many recognition sites for commonly used restriction enzymes. Just now we said there will be very few or single usually. So this one is wrong. Lot of recognition sites generate several fragments which makes gene cloning easy. No, this one is wrong. It won't make easy, it will be complicated. So this one is also wrong. We will go with both or incorrect. 61. If a foreign gene is ligated at BAMH1 site of vector PBR322, then resistance for dash. It will be resistant for tetracycline is lost, ampicillin is lost, tetracycline is not lost, more than one option is right. So here the right option is option A, tetracycline is lost, because the vector PBR322 contains two primarily antibiotic resistant genes that serves as a selectable marker, that is ampicillin and tetracycline. The restriction site for the enzyme BAMH is located in the tetracycline. If for example if I say this is the PBR322, there will be a site for restriction enzyme, that is tetracycline here, and we will be having BAMH here. If a foreign gene is ligated at this site, which is for tetracycline, and there is a site for ampicillin, this is the site for ampicillin. If you are ligating any of the foreign gene at a BAMH site, what happens, it will lose the tetracycline, because this is the area for it is resistant for what, tetracycline 1. If you are ligating at this area, you will be losing what, ampicillin 1. So when he is asking for BAMH, you will lose what, tetracycline. Next, 62th one. There is the recombinants mentioned previous question. Non-recombinants by plating the transformants on tetracycline, planting the transformants on the ampicillin. No, both of these are necessary. Yes, both of them are necessary. Next, 63th question. Recombinants mention in if a foreign gene is ligated at BAMH, one site of vector PBR322 will grow in ampicillin and tetracycline both. Grow in ampicillin but not tetracycline. See, when you are adding your foreign gene at BAMH, which is tetracycline is lost there. If tetracycline is lost, how come it is going to grow on tetracycline, isn't it? But here ampicillin is not at all disturbed. That's what happens. They grow in ampicillin but not in the tetracycline. The right option is option B. Next, 64th one. Non-recombinant transformants will grow in ampicillin and tetracycline both. Yes, of course, because they are non-recombinants. They are not at all recombined to the site. This is ampicillin and this is ampicillin and this is tetracycline. If the foreign gene is not at all inserted either the site, it won't be lost, so it will grow on both in ampicillin and tetracycline. Next, 65th question. Non-transformants E. coli will grow in ampicillin and tetracycline both, grow in ampicillin but not tetracycline, grow in tetracycline but not ampicillin, grow neither in tetracycline nor in ampicillin. So here the right option is option D. 66. When rDNA is inserted in coding sequence of beta-galactosidase, the enzyme gets synthesized, blue colored colonies are produced, colorless colonies are produced, orange colonies are produced. So here the right option is option C. Colorless colonies are produced. This is called what? Insertional inactivation. When any of the rDNA is inserted into the coding sequence of the enzyme, that is beta-galactosidase, it disrupts the DNA, isn't it? This process is known as what? Insertional inactivation. When we are going to insert it, it is going to be inactivated. Because of the gene is that is disrupted, the enzyme beta-galactosidase is no longer produced or else become a non-functional. That's what color change will be colorless. Next, 67. Ti plasmid stands for dash and are present in dash. Ti stands for tumor inducing plasmid and naturally present in Agrobacterium tumefaciens. So here Ti tumor inhibitors, no, tumor inducing, it is tumor inducing Agrobacterium tumefaciens, right? So here tumor inducing Agrobacterium, D is right, because here inhibiting is given, so wrong. Agrobacterium species is given, wrong. Agrobacterium tumefaciens is right, but inhibiting is wrong, so this one is wrong. 68th question. The Ti plasmid being used as cloning vector causes crown gall disease, is not pathogenic, is pathogenic. More than one option is right. Ti plasmid is not a pathogenic. Competent host, from the concept competent host. 69th question. DNA is hydrophilic and can pass through the cell membrane, hydrophobic and pass through the cell membrane, hydrophilic and cannot pass through the cell membrane, hydrophobic and cannot pass through the cell membrane. So DNA is actually hydrophobic and cannot pass through the cell membrane. As you know, DNA is hydrophobic and cannot pass through the cell membrane. That is, when we talk about the interior of the cell membrane is hydrophobic, DNA can't pass through the membrane on its own. That's what happens in biotechnology, treating cells with the divalent cations like calcium ions, and even heat shock method are used necessary to make a host cell competent to take up the recombinant DNA. So for the rDNA technology, those all techniques are very important for the DNA which is not going to pass through it, because DNA is hydrophilic, cannot pass through the cell membrane. Next question, that is 70th question. Bacterial host cell are made competent to take up rDNA by treating with Na plus, no. Treating with Ai3, no. Treating with calcium, right. So here the right option is option C, treating with calcium ions. Next question. Question number 71. Choose the correct sequence to be followed to enable DNA to take up rDNA. First, treating with divalent cation. Next, heat shock. No, it next will be incubating on ice. 1, 3, and lastly heat shock. So here 1, 3 and then 2. 1, 3 and 2. One, 3. Again incubating on the ice. So 3. 1, 3, 2, 1. Sorry, 1, 3, 2, 3. The right option is option B. First it will be treating with the divalent cation, next incubating on the ice, again heat shock, again incubating on the ice. 72. Other methods for introducing foreign DNA into the host cell are microinjection for animal cells, right. Gene gun for plant cells, right. Disarmed pathogens, right. So all of this is right. Next question, 73th question. In microinjection technique, rDNA is injected into cytoplasm, nucleus, cell membrane, lysosome. Here the right option is option B, nucleus. The recombinant, that is rDNA, is directly injected into what? Nucleus of an animal cell using a glass micropipette. 74th one. In biolistics, cells are bombarded with high velocity. A. Microparticles of iron, no. Macroparticles of tungsten, no. Microparticles of gold, yes. This one is right. C is right. Biolistics is nothing but gene gun method. This is the technique used to genetic engineering to introduce DNA into the cells. Specifically we can talk about the plant cells. Next questions from processes of recombinant DNA technology. Identify correct 75th one. Identify correct sequence of the process of the rDNA technology. First, there will be always isolation, transferring of rDNA, no. Isolation of DNA first. So I will write here separately so that we can go with very easily. First, there will be isolation of the DNA. Next, Next will be fragmentation. Fragmentation of DNA by restriction enzyme. So this will be next. 3, 5. Next, isolation of DNA fragment desires. Next is 2. After that, we will going to ligation of the DNA fragment into the vector. That is 6th one. Next, there will be transferring RDNA into the host. There will be transfer. After the ligation, there will be transfer. That is 1. Next, there will be culturing host cells in the medium at the larger scales. That is 4th one. And then lastly, extraction of desired product. 7th one. So 3, 5, 2, 6, 1, 4, 7. So the right option, it seems like C is right. 3, 5, 2, 4, 1, 6, 1, 4, 7. Next, 76th question. From isolation of the genetic material, that is the DNA. Nucleic acid is genetic material of some organisms, no organisms, all organisms without exception, most organisms with some exceptions. So here the right option is option C, all the organisms without exception. Next question, 77th one. How many of the given enzymes involved in the extraction of genetic material from cell of organisms? So here cellulase is given, chitinase is given, lysome is given, ribonuclease, protease and deoxyribonuclease. Among them, enzymes used, that is cellulase is used to break down the cellulose cell wall in the plants, so this is used. Chitinase is used to break down the chitin cell wall in the fungi, and lysosome is used to digest the bacterial cell wall in the bacteria, whereas ribonuclease, it is used to remove RNA from the sample so it doesn't contaminate the DNA. And when we talk about the protease, it used to remove the proteins. And deoxyribonuclease, this is the enzyme digests the DNA. It is like not extract the DNA genetic material, so it is not used as an enzyme that destroys it, so we won't go with that. Given enzyme involved in the extraction he is asking, so this one is destroying. Rather than this, how many? 1, 2, 3, 4, 5 are used. Match the following question. 78th one. Cellulase, it is used in what? Plants. Cellulase enzyme is used for plants. Chitinase is for fungi and lysosome is for bacteria. 1 is 1, 1, 3, 2. 1, 3, 2. So the right option is option A. 79th one. Purified DNA is precipitated out by adding of warm acetic acid? No. Chilled acetic acid? No. Warm ethanol? No. D. Chilled ethanol. So the right option is D. Chilled ethanol. Next one, 80th one. The figure shows DNA separated out, removed by A. Spooning, B. Splooning, C. Spilling, D. Speeling. So this is called what? Spooning. Spooling it is called. SPOOLING, spooling, not spooning. Okay, So here the right option is option B. 81. The precipitated DNA is seen as collection of threads in suspension. Yes, this one is right. Cutting of DNA at specific location. 82. To check the progression of the restriction enzyme digestion dash is used. PCR, gel electrophoresis, DNA fingerprinting, selectable marker chain. So here the gel electrophoresis is used. 83. Preparation of rDNA involves the enzymes specific restriction enzyme, gene of interest, vector DNA, all of this. The right option is specific restriction enzyme. Questions from amplification of the DNA of interest using PCR. PCR stands for 84. PCR stands for polynuclease chain reaction, no. Polylipase chain reaction, no. Polyamide chain reaction, no. So the right option is none of this, because polymerase chain reaction it is. Next question. 85. PCR is an in vitro process, in vivo process, both none. It's a in vitro process. So here what is in vitro and in vivo? In vitro in the sense that is according to the Latin, it is for in glass, means process occurs in the controlled environment, that is outside the living. When we talk about in vivo, it is refers to the whichever the process is there that is going to happen within the living cell or else organisms. This is in vivo, within the organism, this is out of the organisms. Okay. Next, 86th one. How many sets of primers are used in PCR? Only one. One set of primers is required for the PCR process. That is forward primer and then reverse primer. Next, 87th one. Enzyme involved in PCR is DNA endonuclease, RNA polymerase, DNA polymerase, and then DNase. So here the right option is DNA polymerase. So C is right. 88. The enzyme involved in PCR with the thermostability is isolated from Thermus aquaticus fungi. This is incorrect because Thermus aquaticus is not a fungi. Escherichia coli bacterium. But yes, of course, Escherichia coli is a bacterium. It's not a thermostable one, so this one is wrong. Agrobacterium tumefaciens bacteria. This is used in plant genetic engineering, not as a source for the polymerase enzyme, so none of this. So here the enzyme involved in PCR with thermostability is isolated from. What is thermostability? Thermostability is isolated. The enzyme which is used is TUC DNA polymerase, and source of that is Thermus aquaticus, but it is a bacterium, not the fungi. Okay, TUC pol, or else you can also call it as a TUC polymerase, which is a bacteria, not a fungi. So here identify the correct labeling of the sequence. Here the diagram is given. This is the polymerase chain reaction. First there will be a denaturation, next annealing, next extension. Denaturation, annealing, extension. So the right option is C. How can you remember? Trick to remember: DNA heat cool and then build. When DNA opens, we are going to heat it, that is denaturation. Next, we will cool that one, that is called annealing. Again, we are going to build by increasing its temperature. So how can you can short usually you can remember DAE, don't annoy examiner. D for denaturing, A for annealing, E for extension. 90th question. Insertion of recombination at DNA into the host cell or organism. From this portion, 90th question. Ampicillin resistance gene is called selectable marker in case of E. coli is made to take up rDNA bearing ampicillin resistant gene. Yes. B statement. Such E. coli grow on ampicillin containing agar plates. Yes, this one is also true. Both are correct. So we will go with the option. Both are correct. Option A. Next, 91. If a protein encoding gene is expressed in a heterologous host, it is called secondary protein, recombinant protein, transmitted protein, tertiary protein. It is called what? Recombinant protein. 92. In continuous culture system, A, used medium is drained at the end. B, used medium is drained twice in the whole process. Used medium is continuously drained out. None of this. Here the right option is option C. Used medium is continuously drained out. 93. Bioreactors are large vessels. Yes. Used for large quantity production. Correct. Used for biological conversion of raw materials into products. Right. So the right option is all of this. From the downstream processing. 94th one. Downstream processing includes separation, purification, both the above, none of the above. So here it includes separation and also purification. 95. Suitable preservatives are added. Yes, after purification, that is products are formulated with preservatives to ensure the stability and then shelf life. Yes, these formulation need clinical trials. Yes, this one is also necessary. So this one is also true. If the product is a drug, it must undergo strict clinical trials before being going to be released for the human use. And then C, quality control testing is uniform for all the products. This one is not that much accurate because quality control testing and downstreaming procedure vary from product to product. It depends on their nature and then indeed use. So it is not uniform. This one is wrong. So A and B is right. The right option is he is asking how many of the above statement is incorrect. So B is right. 96th question. Optimal condition for the growth include how many of the following? pH, salt, temperature, vitamins and oxygen. For optimal condition for the growth includes pH, salt, temperature, vitamins, oxygen and substrate. So here all of them are needed, so we will go with the option A. Substrate is not given here, but among them they are needed. 97th one. Identify types of stirrer tank bioreactor. Which one is this and which one is this? So here this is stirrer tank. First one is stirrer tank, and then simple stirrer tank bioreactor. This is a cylindrical vesicle with a curved base, isn't it? So this one is sparshed one, stirrer tank. One is simple, this one is simple, and this one is sparshed. Right option is A. 98th one. Identify the correct labels. 1, 2 and then 3. Here it is given. First one, this one is motor. So where is motor? Motor 2 is there, so this option is gone, eliminated, and this one is eliminated. Second one is culture broth, so right option is option A itself. 99th one. Sampling ports are mainly required to keep adding samples into the bioreactors. No. Withdraw small volume of culture. Right. Add acid and base for the pH control. All of this. So here this is right for the bioreactor. Next, 100th one, centuryth question and then last question for the session. Sterile air bubbles are sprayed in the biovectors in a type of bioreactor, that is because air bubbles make it easier to agitate the system. No. Air bubbles increase surface area for the oxygen transfer. Air bubbles enables microbes to grow. None of this. Here the right option is option B. What happens? It increases the surface area for the oxygen transfer. So this is the last question for today's session. If you have any doubt, if you have any queries, please drop your message in comment box. I will reply for that. And please do like and do share my videos. If you like, hit the button. And if you like my videos, if you are compatible with my explanation, please do subscribe. Thank you one and all. Bye bye.